Under this, we have to cover what is plant tissue culture, what is culture media, and how to prepare it. So what is plant tissue culture? Plant tissue culture is a technique to generate clones of the whole plant from cells, tissues or organs on artificial media under the aseptic in vitro controlled environment. So how we are able to achieve a whole plant from a small part of a plant? That's because we have a concept called totipotency. It gives capability to differentiate from one cell into another cell. In today's world, plant tissue culture has a wide range of applications. For example, to obtain a large-scale multiplication of plants, to eliminate the disease, to improve the plant and produce a production of secondary metabolites for that matter. And even we can use this for understanding the constituent responsible for cell differentiation and organ formation. Even though the tissue culture has begun in early 18th century, it was only in 1898 the German botanist Gottlieb laid a foundation stone in the plant tissue culture by culturing the tissues in vitro condition. Now to know more, and even he was also called as father of plant tissue culture. Now to know more about the history of plant tissue culture, you can visit website www.timesofbiotech.com. So what is the culture media? As we know, the plants have organized channel through which it prepares its own food for its growth. But since we are attempting to generate a whole plant from a small chunk of the plant, we have to supplement it with the different growth regulators in the form of artificial culture media. Now, we have divided the components of PTC in 4 parts: inorganic nutrients, carbon source, organic nutrients, hormones. Now, the inorganic nutrients is divided into 2, macronutrients and micronutrients. As the name suggests, macronutrients are added in slightly higher concentration than micronutrients. In fact, the micronutrients are added in trace amounts. Now, what are the examples of macronutrients? The examples are nitrogen, phosphorus, sulfur, etc. And the examples of micronutrients are boron, cobalt, iron, zinc, etc. Now, why we have to add all these macronutrients and micronutrients? That's because if we consider nitrogen as an example, it plays a role in components of protein, nucleic acid, and some coenzymes. If we talk about phosphorus, it has a application in energy transfer, components of intermediates in respiration and photosynthesis. Same way, micronutrients such as copper have electron transfer reaction, play role in electron transfer reaction. Zinc, it has a role to play in chlorophyll biosynthesis, and so on. So we can see that they are very much essential for the plant growth even in the artificial condition. Another component is carbon source. Now the most common source of carbon utilized in tissue culture is sucrose. Since most plant culture are unable to photosynthesize effectively, thus losing auxotrophic ability, they need external carbon for energy. Thus, we are providing sucrose, fructose, for that matter, as any carbon source which will play and which will give energy to the plant. Now, next comes is the organic nutrients. It is divided into vitamins and amino acid. Now, vitamins are required for metabolic processes as a cofactor or a part of an enzyme. The common example would be the thiamine. Now, amino acids, the one of the most common amino acid utilized is glycine. Let's talk about the hormones in detail. Now, these hormones can be utilized either individually or in the combination, depending upon how they have utilized into the culture medium. It will perform its function. For example, auxin give rise to cell elongation, cell division, embryogenesis, organogenesis, whereas cytokines gives rise to shoot buds and cell division in tissue culture. But when they are utilized in combination in specific ratio known as auxin is to cytokine ratio, depending upon this ratio concentration, they will give rise to the different organ formation. For example, if the auxin to cytokine ratio is high, it will give rise to root formation. But if the auxin to cytokine ratio is low, it will give rise to shoot formation. Hence, as we know that these are very much susceptible to the outside environment, they get degraded, we have to use the chemical analogs of it. For example, to utilize auxin, we have to use indole 3-acetic acid or 2,4-D, and as the form of the chemical analog of cytokine as a zeatin. Another hormones which are also utilized in plant tissue culture are gibberellins, abscisic acids. Gibberellins are used for organogenesis, particularly in adventitious root formation, cell division in cell suspension culture. Now, the funniest part is, out of the even though we know 70 plus gibberellin compounds, we utilize only few forms of it. For example, one of those is GA3, which is widely being utilized for the plant tissue culture. Now, talking about abscisic acid, it is utilized for the somatic embryogenesis, particularly during the maturation and germination. Now, let's see how we prepare this culture medium. Now, the preparation will depend upon the types of plant tissue culture we are performing, which species of plant we have utilized, even the part of the plant. Now, so there is no hard and fast rule that we have to use X amount of carbon source or Y amount of hormones. For example, if you can see, these are the formulated media such as MS media, white media and niche media. Here, even these media are not utilized for all type of culture. For example, white media has a low salts and suitable for root culture. But the MS media has high salt concentration and thus it is suitable for plant regeneration from tissues and callus. Coming to the niche media, it has a salt concentration intermediate than the intermediate to MS and white, and thus used for anther culture. So it all depends like what kind of tissue culturing are you attempting to do. You will formulate your own culture medium and preparation, but there are certain steps which are similar for everyone, which are certain steps which we have to take care, and we have to see that we do not do any mistake while performing these steps. For example, we have to only use double distilled water or a millipore water with a neutral pH. That's because if a distilled water or the water which we are using has an iron concentration, it will affect the plant growth. Now, Since it is not possible to weigh each component, as we are going to use it in a trace amounts in a small like 1 mg or even 0.0 mgs concentration, so we have to like prepare a stock solutions. This helps us to maintain the ambient amount of concentration, and thus we have to maintain the accuracy. We have to maintain the accuracy because if any component for any component the media is deficient or we have added in a surplus amount, then it leads to the certain problems like inhibition of protein synthesis, inactivation of enzymes, delayed growth, osmotic pressure imbalance, decrease in photosynthesis, etc. Now, we know that the certain vitamins, not certain, in fact all vitamins and hormones are heat labile. Now, we cannot autoclave it. So we have to filter sterilize it, and we have to add just before pouring the media into the culture plate or culture tubes at ambient temperature of 35 to 40 degrees Celsius, so that a culture media will have a vitamins and hormones, as well as they will not denature it. They will not get heat denatured. Rest of the instruments we have to autoclave, and we have to autoclave the medias which are not heat labile. In certain cases we can use antibiotics, for example for eliminating the contamination, and another example for antibiotic use is for the selection pressure in case of transgenic plants.